Hello everyone, this is Prashant, and in the previous unit we have covered what are the ionic mechanisms majorly inside the cell and how does one neuron combine the information from all the neurons. And that's what and we go through and we have went through the experiments of all the major scientists. Most of them won Nobel Prizes for their discoveries that may appear simple but are actually very complicated. And in this unit, we are going to cover what are the ionic mechanisms between two neurons. And as I stated previously, we are going to discuss one major topic or one major concept that was the start of all of it, the synapse, the gap between two neurons. So let's go. So earlier we know that synapse was found out by Cajal. Basically, he observed uh, the neuron cells inside a microscope and he found out that there is some form of gap between two neurons. But how do we experimentally verify that there is a gap? And this brings us to our next scientist, that is Charles Sarrington. And what did he discover? Okay, so his method was as follows. He dissected uh he dissected a nerve. Okay, cool. And then he electrically stimulated it. The nerve was went through the spinal cord and was connected to the leg. Okay, cool. So we know that when you stimulate a motor nerve, it causes a a reflex. It causes a movement in the leg. For example, if frog's leg or motor nerve is stimulated, the frog legs move. Cool. So okay, that's what he did. He placed electrodes on the sensory and motor nerves, and when he stimulated the sensory nerve, there was an effect in the motor nerve, and he recorded what was the time difference between the two. Instead of one singular axon or nerve, he kind of calculated across the whole arc. Okay, that went through the sensory nerve of the skin, through the spinal cord section, back to the muscle. Okay, cool. That's what he measured. And what did he found out through his reasoning? Okay, uh, through previous experiment, we know that reflex actions are produced in the spinal cord and are majorly the works of spinal nerves that can work independently of the brain. Okay, spinal cord has pair of two types of nerves, sensory and motor, also known as dorsal and ventral. Cool. And we know electricity travels through a nerve and the speed is measurable. Okay, that we know through the experiments of Helmholtz. Okay, Helmholtz. Okay, cool. Now, when we measure the speed across the whole arc, across for the reflex, what we found out is that there is a delay in conduction, and that delay must be due to the gap between the neurons. So, likely there is a different mechanism that is working across the gap between the two uh, neurons or the gap between two neurons, and it must be different from the electrical stimulation that is constant across the whole axon of the neuron. Okay, because the speed is literally uh, different, uh, that is experimentally measured over and over again. So, okay, we know that there is a gap between the neurons that we have verified experimentally, and we can also observe it. And it has some different mechanism than electrical stimulation. Why? Because electrical stimulation takes less time in comparison to whatever mechanism we have between two neurons. And through previous researches or the researches after this, scientists, we also know that their ions are working across neurons. So it's likely it's some form of chemical interaction happening between the neurons that is cause of the delay. But we are going to discuss and prove it otherwise. I mean, we are going to prove it basically, not otherwise. Cool. So the two scientists we have is Lowy and Dahle. And what did they did? Okay, what they did was they put two pairs of heart inside a saline container. There were two containers and there are two heart, and those two containers are connected. And there was a fluid in between them that was kind of salty, like Ringer's solution. And what they did was they took vagus nerve, because vagus nerve connect to heart, and they stimulated the vagus nerve, and they affected and it affected the heart rate not only of one heart but of both the hearts as they measured. And they found out what was the chemical in the vagus nerve that was causing the, what you call it, slowing down of heartbeats. And what did they found out? They found out that it was acetylcholine. So it has an inhibitory function. So their reasoning goes as follows: nerves connect, all the body organs to the brain, different body parts, observable anatomically. And then they found out that electrically stimulating. Oh, okay, fuck that guy, whoever was driving the motorcycle. Anyway, okay. So we know that nerves connect every body part to the brain. Then we also know that electrically stimulating the nerve causes a change in the organ's functioning. That is, it's especially in this case, the heartbeat. Then we found out that uh, a direct contact with the chemicals released affect uh, the organs. Okay, so what we found out that even if two organs or nerves are not directly connected, okay, by any form, only through fluid, the chemicals released by one nerve can travel across through that fluid to another organ, and it causes a change in that organ. And those chemicals that was released by nerves that communicate to the different body parts or give them signal without actually being physically connected to that body part. Okay, those were known as neurotransmitters. So nerves do not have to be physically connected to certain body part. They can send a signal through chemicals. Those chemicals are neurotransmitter, and those neurotransmitters can have an excitatory or inhibitory effect. Okay, and integrating all of that, we came up to the conclusion that is synapse, the gap between two neurons, uses chemical messengers to communicate across neurons on a micro level. This was on bigger level or observable level that we discovered that okay, neurons or neurotransmitters from the neurons of vagus nerve are slowed down the heartbeat even if that heart was not directly connected to that nerve. And now we can see that okay, those chemicals must exist between the neurons as well, and we experimentally verified that as well. So this was their experiment basically. So yeah, heart rate of both the heart is synchronized, and one heart rate slows on stimulation, other also slows even if that is not directly connected to the vagus nerve. So yeah, message or signals are communicated chemically across the nerves or across the neurons. Cool. Makes sense. Okay, so these are the broader classification of all the neurotransmitters as discovered by scientists. So main are acetylcholine, dopamine, norepinephrine, epinephrine, serotonin, melatonin, GABA, glutamate, stuff like that, and endorphins. Those are the main neurochemicals. Okay. Now obviously there is a synthesis of neurochemicals. One chemical converts into another. There's different forms. For example, choline transforms into acetylcholine, tyrosine transforms into dopamine, then it transforms into norepinephrine, and then epinephrine. Okay, cool. So yeah, and we can also observe the structure of synapse under microscope. In light microscope, we can only observe them vaguely, but with electron microscope, we can observe them literally very accurately. And then we can diagram that and label various parts, borrowing from cell theory. That okay, if neuron is a cell, the cell must have a mitochondria. They must have chemicals in them, cytoplasm, stuff like that, some form of membrane and stuff like that. So this is the structure of the synapse. As you can see, there are vesicles, uh, a collection, basically a ball that has neurotransmitters that are generated inside the cell or that are captured across the cell membrane, and they are combined and they travel across the axon on electrical stimulation, and they are released across the synapse, and then they hit the receptor sites in the dendrite of another neuron. Cool. Now we come to our next scientist, that is Bernard Katz and Ricardo Milleri. And what did they discover? Okay, so they found out the role of ions in synaptic transmission. That there was a role of ions. Earlier we know that okay, this is a synapse, and that's how chemical are transmitted and the structure of synapse. Now we can there is one more experiment on ions, uh, ionic moment across synaptic gap. And what they did was they placed electrodes across the two neurons, one on the one side and another on the another side. Okay, cool. And they noted the effect of stimulation of one neuron on another in the form of uh, ele- postsynaptic potential, whether excitatory or inhibitory. And what they found out that concentration of Ca2 plus ions uh, has a major role in affecting the currents that are traveled across the currents that are travel across the neurons. Okay, so how did they did it? Okay, so their reasoning goes as follows. When you remove a Ca2 plus ions or calcium from a solution, what they found out that the potential difference or the synaptic electrical signal across the neuron is reduced drastically. Okay, but when you add calcium ions again, they increase. How do they know that they had to uh, change the calcium ion concentration? Because they have already they knew that Ringer's verified what kind of ions played a major role. And then through discovery, we also know that these ions play a major role in neuron transmission. And now they come up to the calcium. Okay, what was the role of calcium? And we found out that across the axon, the main role is played by sodium and potassium. But across the synapse, the major role is played by calcium. Okay, so by applying chemical methods to test the concentration of calcium across the ion gates, they found out that calcium ion and other neurotransmitters play a major role in communication between two neurons across the synaptic gap. And this is how the process goes. The number one is action potential causes the calcium ions to enter the cell. Those ions combine with the vesicles or the bundle of neurotransmitters, and then they go down and down and they attach themselves to the cell membrane, and then they are released. And there are certain form of proteins that are automatic mechanisms that do this work. So you can see the proteins, uh, calcium ions are what you call it, uh, forcing the vesicles to go down. Then there are latches of what you call it, snares, that T snares and V snare, and they combine with the vesicle and they open the vesicle up after attaching it to the cell membrane. Cool. So this is the overview of the whole process. Okay. Vesicles are released, calcium ions enter the cell. They combine with the vesicles. Those vesicles are attached to the proteins, and those proteins attach it to the membrane, and then membrane open up, and the neurotransmitters go across to either the other uh, cell or through the dendrites or receptive sites on the dendrite, or they are diffused. Okay, or they are what you call it, combined with other cells, or they are thrown away as wasted products across the bloodstream and stuff like that. Okay, cool. All right, we have done that. Now let's go to the different forms of connection between dendrites and synapses. And these are all the possible forms of connections we can observe. Cool. So now that we know that, okay, there are neurotransmitters and they transmit signals across two uh, neurons, and that's how the ionic mechanisms between two neurons happens. Okay, that's the whole overview uh, of the neural transmission. So yep, now we have kind of covered all the things that has to be known about neurons so far in humanity's history, uh, from all the experiments that were done on brain and the brain functions and how what role do neurons play and stuff like that. Now we have know whatever happens in neuron. Now we are gonna cover. Okay, now we are gonna cover besides neurotransmitters or neurochemicals, what other chemicals are there that affect the body? Okay, and spoiler alert, they are hormones, and we are gonna see, okay, how do hormones affect the behavior and how do they interact with our nervous system and stuff like that. And at the end of it, we will discuss all the major research methods or measuring techniques of nervous system that we have discovered so far. And those will be our next two units. One will be hormones, the other will be research methods. And thank you for watching, and see you in the next video.